Merhaba, hoş geldiniz. Yeni bir Nano Open'la karşınızdayız. Bugün misafirimiz Harvard Üniversitesi'nden Doktor Nur Mustafaoğlu. Yakın bir zamanda da Sabancı Üniversitesi FENS'te çalışmalarını izleyeceğiz. Sözü kendisine mi vereyim hocam? Siz bir şey söylemek ister misiniz?
Kendisine verelim. Belki biraz daha ayrıntılı tanıtırsınız kendinizi. Ondan sonra da dinleyelim. Vakit geçirmeyelim çok fazla. Teşekkür ediyoruz.
Evet. İsterseniz ben sunumu açarak başlayayım.
Tabii.
Hello, everybody. My name is Nur Mustafaoğlu, and today I will be talking about bioengineering, diagnostic, therapeutics, and transport applications. And first of all, I would like to describe myself a little bit, introduce myself. I graduated from molecular biology and genetics from Istanbul Technical University, and I did a double major in physics engineering at the same university. Then I moved to nanoscience and nanoengineering, again at Istanbul Technical University. During that time, I actually awarded by Fulbright scholarship, and I had a chance to move to United States to get my PhD education in bioengineering. I went to University of Notre Dame and did all my PhD studies in there during 5 years. And after that 5 years, after I receiving my PhD, I moved to Wyss Institute for Biologically Inspired Engineering at Harvard University. And I did my postdoctoral studies there for 3 years. And now I'm in the transition phase to joining as a faculty member, interdisciplinary faculty member in Faculty of Engineering and Natural Sciences at Sabancı University. And I'm very excited today to talking about my PhD studies and postdoctoral studies and my transition to the Sabancı University. So, as you can see, my road for the education is mostly inspired by the natural and the health sciences, and always my motivation was like, how can I help other people and improve their health conditions by using simplistic approaches of sciences, and then it really meets with the definition of the bioengineering, which is defined as a discipline that applies engineering principles of design and analysis to biological systems and biomedical technologies. And the definition by the University of Washington is actually like what drives me and meets my motivation much better. It defines the bioengineering as a devices innovative solution to open-ended, unmet challenges in biology, health, and medicine. So I got my education from physics, from molecular biology, and during my PhD, I received a lot of chemistry education as well to build a skill set and tools to study bioengineering and help people for getting solutions to their health and medical conditions. So today I will be describing some of these challenges and unmet challenges and open-ended questions. One of them is the biosensors. As you all know, the biosensors are very important devices, and I will be focusing on the immunosensor-based applications of the biosensors. They use a lot of proteins to screen pathogens, disease detections, point of care, clinical analysis, medical diagnosis. But there is a problem with this especially if you are in the nano sizes, and they use proteins to capture the biological components from the biological samples such as blood, semen, and other biological samples to detect the diseases or for diagnostic applications. You have to have these these proteins orientedly immobilize on the small scale of the surface area, and this is a challenging problem, and bioengineering tries to find a solution to these problems. And one of my PhD projects involved to get an answer for that, and I used a method called UVMBS, and I will be describing it in a minute, actually. Before moving to that problem, for biosensing applications of for diagnostic applications, antibodies are one of the mostly used proteins to capture the biological samples, because of their specificity and affinity to their biological components that we call antigens. So they can be used for diagnostics, therapeutics, diagnostics, and actually there are a number of monoclonal antibodies that are improved in therapeutic applications, increasing every year, and the market prices of these monoclonal antibodies are becoming like hundreds of billions of dollars. And it is very important component in these applications, and most of my studies also using antibodies. So this is actually, I don't know if you can see my cursor here. This is a typical structure of an IgG molecule, immunoglobulin G, one of the isotypes of the antibodies. It has two antigen binding sites. It's the dimers of a dimers protein. So it has like two symmetrical structures, and it has two antigen binding sites and two antibody binding fragments. And I will be, this was just a reminder of the structure. I will be describing one unknown region in the antibodies, which we call nucleotide binding site. It conserved in all antibodies. It is this site has two tyrosine in the variable region of the light chain, and one tyrosine and one tryptophan in the variable region of the heavy chain. And these nucleotide binding site has an affinity to the small molecules, aromatic small molecules such as indole tributyric acid, tryptamine, naphthalene, and other things. And we thought that this binding site, if it's conserved in all antibodies, we can use these region of the antibodies to site-specific functionalization, to oriented immobilization of the antibodies. We can also use them for allergy inhibition and antibody purifications. And there are a lot of studies on that region and on these applications. And today I will be describing some of them. So one of them is the Fab Fragment Immobilization. As I described before, Fab fragments are part of the antibodies. They still have the antigen binding site and also nucleotide binding site. So we can still use the Fab fragments of the antibodies to detect some of the biological components and for diagnosing some of the diseases. And I will be also describing using of Fab fragment immobilization, also whole antibodies on different surfaces like solid surfaces, and also on the nanoparticles. And there are also other applications for nucleotide binding sites, such as antibody purification, that we can get like cost efficiently and easy and repeatable and consistent results for purification of the antibodies, which is actually another challenging problem in the field, because as I mentioned, the cost of the antibodies are increasing a lot, and the market for the monoclonal antibodies for the therapeutic applications are huge, and there are a lot of demand for reducing the cost of the antibodies, and we would like to use and develop new techniques that are and improve the conditions for purification of the antibodies. But today, for the sake of the time, I will be skipping the antibody purification applications. Instead, I will be describing another challenging problem in the bioengineering field, describing the blood-brain barrier and how we can deliver drugs to the blood-brain barrier, and what are the options for blood-brain barrier models, and the more challenges in that system. Okay, first, let's start with the Fab Fragment Immobilization technique. As I described to you, the Fab Fragment has both antigen binding side of the antibody once we cleaved it from the whole antibody, and it also still includes the nucleotide binding site and the conserved region in all of the antibodies. And as I also described, small molecules such as indole tributyric acid here has a moderate binding affinity to that side. So if I synthesize a molecule such as like that, indole tributyric acid, a length of the linker, and also some functional moiety, in this case a biotin here, I can conjugate this Fab fragment with this molecule, and using I used a UV exposure here to covalently cross-linking it, and then I can easily mobilize this biotinylated Fab fragment onto a strep tablet encoded place, because using the big affinity between the biotin and strep tablet. So I can obtain orientally immobilized Fab fragments on the surfaces that can be used for detection of the biological samples, antigens in the biological samples. So to demonstrate that, we first needed to answer, okay, I synthesized a small molecule and I have the Fab fragment, and once I conjugated them, to conjugate them, actually, I need to use a UV energy, and I need to make sure I'm conjugating the molecule to the Fab fragment in 100% efficiency, and I also need to make sure that I'm not destroying the protein, the Fab fragments, while applying the UV energy. So we tested those conditions and identified the best optimal conditions for exposing the UV energy, and we found that 1 joule per centimeter square in this case for this antibody, which are anti-prostate specific antigen antibodies, are the most optimal conditions. And then we compared this functionalization method with the other traditional functionalization methods like physical absorption, conjugating antibodies with NHS biotin chemistry, or conjugating antibodies with the primary amines of the lysine amino acids that exist on the Fab fragments. And we found that the UV-NBS method provide us the most efficient method for Fab fragment immobilization. And also, once we introduce the antigen to the immobilized surfaces, it provides us the most oriented version of it as well, because we were able to detect the antigen in the most sensitive way using the UV-NBS method. So to summarize this part, with the site-specific photo cross-linking of the Fab fragments with UV-NBS methods, we were able to get very high Fab fragment stability and antigen binding activity of it. And we were able to immobilize Fab fragments using this with enhanced levels of immobilization of Fab fragments, higher antigen detection efficiency, and improved antigen binding sensitivity. Actually, we show that this system works for different antigens and for different antibodies, and we detected prostate specific antigen. We also detected Ebola antigens in different systems. Then this was the application of oriented immobilization on solid surfaces. So if you have a very small area of of your biosensing device, you can increase the antigen binding sensitivity by applying by actually immobilizing more antibodies on the surface. Then the next question was, can we do it on mobile surfaces to be able to apply or use these diagnostic systems and increase the sensitivity of the diagnostic systems for microfluidic devices or paper-based devices? So we tested this immobilization method on nanoparticles, and we use gold nanoparticles. This time, instead of synthesizing a molecule with biotin, we synthesize a molecule with tile functionality at the end. So once we conjugate the antibodies with this linker, we were able to get tilated antibodies. Then they can easily immobilize on the gold surfaces, such as gold nanoparticles here. And then we actually stabilize the gold nanoparticles so to eliminate their aggregation. And to hope that, since they are more mobile, they can be used in many different applications as well. So to demonstrate that they are functioning and they are conjugation and immobilization on the surfaces, we used two different antibodies. They are both against the prostate-specific antigen. Both of them are monovalent antibodies, so they recognize only one epitope on the prostate-specific antigen. And once you conjugated these antibodies on the gold nanoparticle surfaces, the antigen binding sites of these antibodies will recognize different epitopes, and they will form a sandwich on the antigen, and they will create an aggregation. So I can detect these aggregation using a dynamic light scattering approach, and I can determine the concentration of the prostate-specific antigen in the biological samples. So we also did a lot of control experiments and also the comparison experiments, but at the end, the final result with the actual biological samples, the spiked prostate-specific antigen in human plasma provide us very sensitive results with the lower limit of detection of 4 picomolar. And as you can see here, if you have only one type of antigen in the system, you will not be able to get the aggregation of the nanoparticles, because they will form only an antibody and an antigen system, and they will not form any aggregation. If you have both of the antibodies together, they will form a clustering formation because of the bivalency of the antibody recognition. And you will get clusters and so larger and larger clusters if you increase the concentration of the antigen in the system. So this was also very interesting finding, and we demonstrate that the UV-NBS method can be utilized to generate antibody functionalized gold nanoparticles, and that results yield significantly enhanced antigen detection efficiency and improved antigen binding sensitivity with high specificity. So, and the lower limit of detection was also in the range of the defined range by the National Cancer Institute. And this was also, it is also very important to indicate that this was only a one-step detection method, because if you already prepare the conjugation of antibodies, all you need as the biological samples derived from the patients, and you can get the results within like 5-10 minutes. So it is also very important to indicate that in that sense. So so with the UV-NBS method, yes, UV-NBS region on the antibodies, we actually worked out, and there are still a lot of work to be done in that area. As we can do the conjugations, improve the conjugation systems without using the UV, because we still know, like each time when you are conjugating an antibody, you have to define an optimal UV conditions, and you need to work out the optimization for each antibody specifically. And this area on the antibodies can be used for conjugating drugs to the antibodies and create antibody drug conjugations for the therapeutic applications. And since UVNBS is located in the variable region of the antibodies, even the smaller parts of the antibodies can be used. And there is a high demand for the finding new antibody ligands. And of course, there are the approaches to get like using the nucleotide binding site and to provide the antibodies and creating affinity columns for the antibody purification problem. So and these are only the using the nucleotide binding site. There are also a lot of biosensing applications that can be used for the oriented immobilization techniques, and also other approaches and maybe different detection methods can be done, and the biosensing applications can be improved a lot. So, and today I will be also like these were part of my PhD thesis, but I also worked one different problem, which is blood-brain barrier model. This was specifically interested was an interest of me because I was curious about brain all the time, and I wanted to know like why we cannot cure Alzheimer or Parkinson. What is the challenge behind those, why we don't understand many of the functionality of the brain. So blood-brain barrier was one of the challenging part that behind of the non-cureness of these diseases, which is a protected barrier, protecting barrier of the brain. Actually, the endothelial cells in the brain capillaries are specialized in a way that they really form very tight barriers, and the junctions between the cells are between the endothelial cells are very, very small. They are in Armstrong levels, and which is very different than the other organ capillaries or also vasculators. And so this is a beautiful structure, a schematic of the blood-brain barrier. You can see the yellow reddish color are the endothelial cells. They are surrounded by the pericytes, and the end feet of the astrocytes are also reaching to the endothelials, so they can connect between the neurons and the endothelial cells to create the homeostasis of the brain. So as I described, the blood-brain barrier is protecting the brain. So it has a very complex structure. This is a cross section of the blood-brain barrier. You can see the lumen here, where the blood flows. It has the endothelial cells surrounded by it, and there are a lot of tight junctions expressed on the blood-brain barrier, which is the expression levels of the tight junctions, and at an adherent junctions molecules are extremely high in the blood-brain barrier compared to the other organ endothelials. And there are also a lot of efflux proteins expressed on the brain endothelial cells compared to other endothelial cells in the human body. And that helps to complex and dynamic structures, creating a dynamic structure of the blood-brain barrier, which regulates the entry of the blood-borne molecules into the brain and protects the brain from pathogens and toxins, and also preserves ionic homeostasis within the brain microenvironments. But on the other hand, it becomes a restrictions for the drug delivery, because most of the small molecules, actually over 98% of the small molecules, cannot cross the blood-brain barrier they are effluxed and pumped back. They are taken by the cells and pumped back to the blood circulation, and which makes a challenging problem because we cannot dose drugs if we have a disease in the brain. So we cannot have enough concentration of the drugs in the targeted disease regions in the brain. So the challenge here, like how we can trigger or trick the system by creating engineered systems such as liposomes, antibodies, other nanoparticles, or physically opening the blood-brain barrier without damaging it a lot to deliver the drugs to the brain in order to be able to cure the brain diseases. So during my PhD studies, I focused on designing liposomal approaches. To be able to do that, I synthesized lipid structures which will have peptides, which is targeting a protein in the blood-brain barrier, a protein expressed a lot in the blood-brain barrier compared to other brain endothelial cells. And I formed liposomal structures like this, and this will, if I can target the brain endothelial cells, and if I can enter these liposomal structures into the brain, this will enable me to deliver other therapeutic regions into the brain side too. The idea was that, and I tested different types of peptides that are targeting different proteins on the brain endothelial cells. Some of them did work, some of them did not work well, and this is just like different types of peptides that are targeting different proteins on the brain endothelial cells. Some of them were binding and also uptaking well. Some of them were binding but not uptaken well by the cells, by the primary brain endothelial cells. And then next, I chose one of them that worked pretty well, and try to form different formulations of the nanoparticles with different sizes, with different linker lengths. So in order to be able to play with the chemistry and also to increase the enhancement, basically enhance the uptake of these nanoparticles by the endothelial cells so they can enter the blood-brain barrier in an efficient way. So, and some formulations worked better than the other ones. And also I played with the concentrations of the peptides on the nanoparticle surfaces, and found that like some of the concentrations are actually working a lot better than the other ones. But there was a problem when I started testing those formulations that are created in the in vitro systems, not only the cells, like forming a blood-brain barrier model in a transplant model. I found a problem that like actually I cannot form a very tight barrier formation in these 2D cultures. And I was using primary brain endothelial cells. Basically, this is a schematic of the transwall system, which has like traditional plates and inserted by a compartment which has a porous membrane at the bottom. So you can actually see the endothelial cells at the top to form the blood-brain barrier. And once you dose your samples on the top, if they are formulated well and targeting the brain endothelial cells, uptaken by the brain endothelial cells, you should be able to collect some of the samples at the bottom compartment. But my finding was I was not being able to form very tight barrier formation with these cells and with this model as well, because this model also enables us to check actually if I can form the blood-brain barrier. And it was very challenging because if you don't form the blood-brain barrier efficiently, whatever you are dosing, they may be crossing the blood-brain barrier or the system with diffusion or by specific concytosis mechanisms. So I wasn't sure which one is actually occurring in this when I'm testing my different formulations. So I decided like what else I can which other models I can use, and I looked through the literature like what other people are using for testing these things. And of course, first of all, it was the cells that I was also doing, and I tested the initial systems, and they have very high throughput and low cost, and they are easy to handle, but they are lacks the complexity and a poor relationship to the in vivo. And another thing is using the animal models. They are very complex and they provide optimal cell-to-cell contact. But on the other hand, they are very variable, and of course, mouse data is not equal to human one, and we would like to cure the human diseases, not the mouse diseases or any other animal diseases. So, and organoids are also emerging rapidly in the field right now, and they are self-organized systems. Of course, they are creating an optimal cell-to-cell contact, and they can be plantable, but they are hard to replicate, visualize, and really my purpose for this study was selecting the system, screening the formulations that I created in the lab, but the organoids did not allow me to do it. And the last system was the microfluidics. Using the microfluidic organ-on-a-chip systems, you can actually create optimal barrier function. It's a high contact systems, but it's not a high throughput system, and it may become very expensive. But for my purposes, it was the best match for my for selecting of the drugs, because I can still approach to both brain and the vascular channels, and I can screen the drugs in an efficient way. So I choose this system for my further studies, and in order to be able to learn these systems, I moved to Harvard to Dr. Donald Ingber's lab, who is a pioneer in the organ-on-a-chip systems. And in his lab, there are like more than 20 different organ models using the microfluidic approaches, and they generate a lot of different disease and healthy models of different organs. So this is the model that I helped generating in the Ingber lab, which has two microfluidic channels, which are separated by a porous membrane. At the bottom channel, we seeded iPSC-derived, like these are induced pluripotent stem cell-derived brain endothelial cells. And also on the top of the porous membrane, we seeded the astrocytes and pericytes. So the bottom channel mimics the vasculature of the brain and blood-brain barrier, and the top channel mimics the brain side of the blood-brain barrier. So in order to be able to generate very tight barrier formation with the endothelial cells, we use an approach for differentiation of the endothelial cells in hypoxic conditions to mimic the embryological developmental conditions. And we differentiated the cells in hypoxic conditions and seeded them on the chips after differentiation, and continued to culture them with the astrocytes and pericytes in a while before running any experiments with them. And with this model, we were able to see actually these cells are expressed tight junction proteins such as ZO1, and another tight junction protein, which is known to be expressed in brain endothelial cells, specifically Claudin-5, other brain endothelial cell markers, PKM1, or transporter markers, GLUT1, and efflux markers, PGP. And also we observed beautiful tight junction formulations and direct junction formulations using electron microscopy with these models. And then we compared these model with the, like, what if I use primary brain endothelial cells I was using in the trans walls in these chip models. Of course, I still did not get any barrier integrity, which was still a problem. And then if I use these endothelial cells in the trans wall model, which is a static model, can I still get high barrier integrity? And the results were showing, okay, I can get very high barrier integrity at the beginning, but over time, since I was not able to apply shear stress and flow conditions in the static model, the barrier integrity drops over time. However, with the chip model, when I seeded the endothelial cells, astrocytes, and pericytes in the model, I started applying the flow conditions and shear stress to the model, mimicking the physiological environment in the microfluidic device. I was able to cap the barrier integrity over 2 weeks. And we also checked the functionality of these models to see if the efflux proteins, we observed that efflux proteins are expressed, but we wanted to make sure that they are functioning as expected in the physiological conditions. And we tested some substrates that are known to be to substrates, and the PGP, which is a polyglycoprotein, a non-efflux protein on the brain endothelial cells. And once we inhibit the PGP function, they were able to cross the blood-brain barrier. But if we don't inhibit the PGP function, they were not able to cross the blood-brain barrier. And we compare these results with the trans-wall system. And the chip model with the flow and the shear stress were able to recapitulate the phenomena that are known in the physiological conditions with the in vivo conditions. And also, like, the next question was, we built this system, can we use it for screening of the drugs, or can we use it for testing different drugs? And there are different ways of crossing the blood-brain barrier: paracellular aqueous pathway, transcellular lipophilic pathway, transport proteins, absorptive transcytosis. But the ones that we were more interested in, receptor-mediated transcytosis, because it is the most invasive way. So it will not disrupt the blood-brain barrier when we are applying this applying the drugs into the system. So the first approach was, can we actually intentionally open the blood-brain barrier physically and reversibly? This is the system that has been using in the clinicals today, actually, and doctors are applying to patients with hypertonic solutions such as mannitol to open their blood-brain barrier and inject the drugs for a certain period of time, and then after stopping the hypertonic solution exposing, and the blood-brain barrier reverse it back, so it forms tight barrier formation again. And we recapitulated that phenomena and observed the blood-brain barrier opening over time after exposing the hypertonic solution, so that the barrier integrity drops. At the same time, we started injecting larger molecules into the model, so we can recognize the samples, the molecules, and antibodies, which are therapeutic agents for the cancer, crossing the blood-brain barrier. And later, when we stop exposing the hypertonic solution, we recovered back the barrier formation and decreasing the concentration of these molecules and drugs. And the next, can we do it? The next question was, can we do it targetedly without physically opening the blood-brain barrier and using the proteins that are expressed on the brain endothelial cells a lot more than the other endothelial cells. And we targeted, we use angiopep2, which is a peptide that are targeting LRP. And we also use anti-transferrin receptor antibodies to test these phenomena and to be observed that they can actually cross the blood-brain barrier in an efficient way and compared to their controls. And then this is a summary of the BBB chips assays. We were able to establish a physiological blood-brain barrier model in vitro, which recapitulates in vivo microenvironment with oxygen levels and shear forces. And that provides a stable barrier function, express high levels of efflux proteins. And that also provides us a multiplex system for screening of therapeutic agents for with medium throughput. And we also collaborated with collaborators from the Boston's Children's Hospitals. They were actually studying extracellular vesicles crossing generated by the brain-seeking breast cancer to going to the brain, and their mechanism, try to understand their mechanisms, and they were using different in vivo systems and in vitro systems. And we, this is actually one of the studies that the data was correlated with the in vivo systems, mouse and the zebrafish models, and our in vitro model for the blood-brain barrier on a chip model, and show that actually the brain-seeking extracellular vesicles generated crossing the blood-brain barrier and targeting the astrocytes. So think I lost you in a minute.
Hocam duyuyoruz ve görüyoruz slaytınızı.
Harika. Bu son slide'ım. This is my last slide. Actually, is a summary of the blood-brain barrier. There is it's still a challenging area, and there are lots of unknowns, and there are lots of ideas around it to study. And like with the models, with in vivo models, with in vitro models, and there are a lot of needs to identifying novel targets with the for the brain endothelial cells to target with the proteins or nanoparticles to cross the blood-brain barrier. And there is a of course, there is a high demand to and need for the in vitro models, because we cannot always study human brain in vivo conditions for many reasons, and the biggest one is the ethical reasons. And of course, once we build the healthy models, in vitro models, we should also study the disease models, Alzheimer and cancer models. We should also study the patient-specific models, incorporate more complexity into the systems, like incorporating neurons, other brain cells into the systems, and to create even more physiological conditions. And there are a lot of bioengineering approaches and drug designs that can be used for targeting the blood-brain barrier. so a lot of ideas in that system, and I'm hoping to move some of these projects to Sabancı University and build my lab at FENS and study some of these projects and collaborating with the researchers at Sunum and FENS and in general in Turkey and Europe and America to basically to develop systems that we can cure the brain diseases. And with that, I can actually thank all of my colleagues, collaborators, PIs, from the Wyss Institute at Harvard, from the University of Notre Dame, and other collaborators from the University of Wisconsin-Madison and Brigham Women's Hospital. And thank you for your invitation and giving me the chance to present my research here. And I'm happy to answer if you have any questions.
Hocam biz teşekkür ediyoruz yoğun programınızda zaman ayırıp sunumdan Open serisinde bizlerle olduğunuz için. Soru var mı acaba? Biraz bekliyorum ama belki bir cesaretle birisi soru sorar diye. Have you any questions?
Maybe I could ask you a question. I'm Ali Koşar. I think Nur also I think knows me.
Of course.
Just
hello.
Maybe, out of my curiosity, you showed a chip, right? It really seems to be a standard chip. Can you elaborate more on its design and if it has any kind of specific feature? Can you highlight it?
Oh, of course. As you said, it's very standard model. So it has, and it's also actually very simple model as well. It has just two channels and a porous membrane, and it both the membrane and the model is made of PDMS. And
Are the dimensions,
actually,
for example, in micro scale, right? Micron size, 50 microns, something like this, right?
Yes, these are actually like huge channels. They are not in nanoscale. They are definitely micro scale, and the top channel is actually slightly taller than the bottom channel. The bottom channel is at 200 micrometer, and the top channel is at 1,000 micrometer. They are big. And maybe I can actually. So this one is very standardized chip model, which is also sold by a company that is spin out from the Dr. Inkber's lab called Emulate. And we also used in-house chip models that incorporated electrodes in the chip model to be able to measure the barrier integrity. So they look like this one. So once we see the cells, we can get the electrical resistance readings, like basically their impedance readings to be able to calculate.
Oh, you have thin films, right, at the back. That's how you read it?
Sorry?
You have thin films, right, at the back. So you read from there?
Yeah.
Mm-hmm. Yeah, thank you. Mm-hmm.
Başka sorumuz var mı acaba? Have you any questions? Have other? Evet, chat kısmında da sorumuz yok. Hocam çok teşekkür ediyorum.
Teşekkür
Etkinliğimizi
ederim.
sona erdiriyorum bu durumda. Tüm katılımcılarımıza da çok teşekkür ediyorum. Hocam sizi aramızda görmekten de çok mutlu olduk. Mutlaka sunuma da bekliyoruz. Herkese çok teşekkür
Teşekkür
ediyoruz.
ederim.
Teşekkür ederiz.
Eğer sonradan soru sormak isteyen varsa bana elbette ulaşabilirler mail yoluyla veya diğer yollarla. Sorularınızı cevaplamaktan memnun olurum veya tartışmak istediğiniz bir şey varsa benim sunumumla alakalı veya diğer research'lerle alakalı onlar için de iletişime geçmekten memnun olurum tabii ki.
Çok teşekkür ediyoruz.
Ben teşekkür ederim. Sağ olun.
Görüşmek üzere haftaya başka bir Nano Open'da. Görüşmek üzere diğer katılımcılarımıza da. Hocam çok teşekkürler tekrar. Hoşça kalın.
Hoşça kalın. İyi günler.
İyi günler.